Okay, maybe a couple, 3, 4, 5. Okay, good. So I'm going to go ahead and summarize what this paper is about anyway, just to get everybody on the same page. Sorry, by the way, I'm Deepa Agashe. I'm one of the organizers of this meeting. I'm sorry I couldn't be here today morning. Still a little bit sick and low on battery, so we'll have to see how far my battery runs and try and finish this discussion before I'm totally gone. So this paper is one of these really, really nice early examples of what's called adaptive radiation, right? So now I know that some of you at least don't have a biology background. So you may not have understood half of this paper, which is perfectly fine. What we'll try and do is go over some of the main concepts and terms so that you are sort of catching up with everybody else. There's not too much math involved here. So the biology people are probably fine in some sense. Yeah? So does anybody have any idea what is adaptive radiation? That's one of the first things that may not be obvious to half the crowd. Everybody fine with adaptive radiation? You're not quite sure? You're fine, okay. So do you want to explain quickly what it is?
So if there is a particular species which have a selective advantage, so the chance, because of that selective advantage or adaptive advantage, that it will radiate into much more subspecies.
Not quite there, but okay. So the basic concept is, so what does the word radiation suggest to everybody, right? That is, there's a spreading out, right? So that's kind of part of your definition. And there's a spreading out, but the way we define it is that it happens in a very relatively short term, okay? So the way we think of speciation, if you remember the trees or the phylogenies that many of you may have seen, has anybody not seen a phylogeny ever? Okay, good. Everybody's has some, no. You got to speak up because otherwise I will keep going thinking that you know this. So a phylogenetic tree is effectively just a basic tree shape with branching, and effectively what it denotes is evolutionary relationships between taxa. Okay? So you can imagine, for example, there are humans here, there are chimps, right? And other ape species. So we know that we are most closely related to chimps. We share a very recent common ancestor, which is in some sense fictional at this point because we don't know exactly what this common ancestor looks like. But there's this sort of binary splitting that we think of when we think of trees, when a species gives rise to two new daughter species, right? And the point is, so these are all species that exist currently. If we keep walking backwards in time, eventually we'll end up with the last universal common ancestor of all life forms that we see on Earth right now, right? And so this is effectively a phylogenetic tree, which represents the phylogenetic relationships between taxa, between groups, between species. It's up to us to define what these endpoints are. Okay? These may be populations in some cases of the same species. So we may think of, for example, tiger populations from India versus other parts of Asia versus Siberia and so on and so forth, right? Or we could think of different species. We could even think of big groups such as animals and plants and so on and so forth. It's up to us to define what we're exactly putting these groups as. So is everybody okay with that? Yeah. So effectively what we're trying to say then is if there's a radiation of new species that has come up in a relatively short time, so imagine this phylogeny now looks like this, where a lot of species have evolved in a very short time, such that we can't even tell precisely, we have very poor resolution here, okay, of being able to say exactly which ones share the most recent common ancestor and which two are splitting from which precise one ancestor, yeah? So this kind of situation would be what we would call as a radiation, okay? And if this radiation is driven by adaptive processes, we would call it adaptive radiation. Yeah? Is everybody okay with the concept of what is adaptation? Yeah. You could have allopatry, right, which is, for example, a chance event, a series of chance events. Let's say there's a big river that splits into multiple small rivers due to some geological upheaval. And each of those small rivers because they are now separated populations that don't exchange genes anymore for a long time, even though they may not be adapting necessarily, they may still eventually over time accumulate enough differences genetically that they can no longer exchange genes, without those differences being driven by adaptation to each of their smaller rivers. Does that make sense? Does that make sense to everybody? No? You're still skeptical? Huh? Where's the radiation? If it happens in a short time, where each of those rivers, the fish populations in each river accumulates mutations in a relatively, now it's a bit dicey, it's a bit subjective. What is my radiation may not be your radiation, right? So there are issues there. But the point is that in general, it happens in a very relatively rapid rate, and there's lots of species formed within a short time period. I'll give some examples of this in the next slide. Yeah. Allopatry and radiation. No, so allopatry refers to the distribution of species. Okay, so let's say there's a population. So humans, for example, we have sympatric populations of humans where, maybe humans are a bad idea. Okay, so let's say fish, for example, in two different rivers are necessarily allopatric if the rivers never meet each other, right? Two distinct rivers, two separate continents. Any population on Asia and North America are going to be allopatric, whereas sympatric means you're living in the same area. Yeah? So any species that are, for example, spanning the divide of India and Pakistan border will be sympatric species. They have the possibility of exchanging genes, whereas in allopatry, there's no possibility of exchanging genes. Okay, is everybody fine with this? Any skeptics? Okay, good. So the basic idea is adaptive radiation is thought to lead to a lot of new species forming. So of the hundreds and thousands and millions of species we know on Earth, it's thought that adaptive radiation may have led to quite a number of them, right, arising in a short time frame. And so question has been, what drives this adaptive radiation? What kinds of selection pressures drive adaptation? Under what circumstances is adaptive radiation more likely to occur versus non-adaptive radiation, and so on and so forth. Okay, so people have been interested in these questions for a long time. And this paper is one of the few times that people have suggested this happens in a very short time, in a week, for those of you who have read the paper, right? And in a microbial population, because nobody had thought about microbes as being really interesting for speciation work for a long time. So let's dive into this paper. I want to give you a little bit of background first. So here are the promised examples of adaptive radiation. Many of you have heard about the Galapagos finches, right, Darwin's finches, on the Galapagos Island. What we know now from decades of research is that there was an original single species of finches which colonized the island, and over time gave rise to a large number of species within a relatively short time span, each of which is specialized, adapted to a different food source. Okay? So their beak shapes and the rest of their body shapes, allometry, the head shape, all of that has been shaped by what kind of nuts or seeds that they are specialized to eat, or insects in some cases as well. This is an example of adaptive radiation. Here's another very famous example of adaptive radiation, which are the cichlid fish. There are these 3 different lakes in Africa, in this area, which have been intensively studied for, again, many decades. Here's a phylogeny. It's similar to the tree I drew for you earlier. And you can see these branches are all very short, right, relative to these larger branches, which are fish from other parts of this continent, right? So if you imagine the distance of these lineages telling you something about how long ago the branching happened, then this branching has happened relatively recently. So this is a radiation, and we know it's adaptive. It's given rise to this crazy sort of diversity of shapes and colors and sizes and so on, because all of these different fish have adapted to use different niches in their lake habitats. Okay, so within each lake, there are some fish which are bottom feeders, which will feed sort of on algae which are attached to rocks at the bottom of the ocean. Some are sort of specialists at going in crevices between rocks. Some are predators, some are top-down hunters, some are bottom-up hunters, and so on and so forth. You get the idea. And so their mouth parts and the rest of their body has been shaped by these specializations in a very short time. Again, this is something that a lot of people have studied, and beautiful work also showing what is the genetic basis of some of this radiation, and so on and so forth. Okay? So in today's example then, we're dealing with this bacterium called Pseudomonas aeruginosa, and it's a common bacterium. It's a facultative pathogen, which means that given the right chances, given the right host, such as humans who are compromised in terms of their immune system, can colonize our lungs and cause disease. But in healthy humans, it can't invade, right? But this pathogen has been a nice model system used in the lab to understand a lot of different aspects of how bacteria evolve and what their ecology is and so on and so forth. So what they're showing here is when you take a beaker full of, and seed the culture, seed the beaker which has nutrient medium with this Pseudomonas original strain, which is called smooth, okay, the colony on an agar plate, which is formed by this ancestor, is this nice smooth round colony. Has everybody seen bacterial colonies on agar plates? Anybody who has not? You haven't? Okay, can you remind me? I'll bring some tomorrow so you can see, because otherwise it's just sort of hard to imagine what I'm talking about, okay? But effectively, these are small petri dishes where there's a gelatin, you had jelly, right? Okay, so it's a jelly-like material in the bottom. We put some nutrition in it, and the solid surface, when we put single bacteria on it, they will grow and form these colonies. So each such colony consists of lots and lots of cells, okay? And this is now visible to the naked eye, but a single bacterial cell is too small to be seen, right? So we can effectively know that each bacterial colony was most likely seeded by a single cell at some point. Is that okay with everybody? Yeah? Okay, so I'll get some colonies for you to see tomorrow. So, we start with the smooth guy, and we let the broth sit for a long time, about a week, and bacteria divide very fast typically, right? These guys do. In a few hours, they'll keep doubling, right, replicating. So even though it seems 7 days is a short time, it's actually quite a lot of generations for these bacteria. And you find that they diversify into these 3 forms in these 7 days. There's a form called a wrinkly spreader, WS, which, as the name suggests, is wrinkled and spreads, right? It's not a sort of dome-shaped colony like the smooth guy. And there's a second guy, which is fuzzy spreader, which again, as the name suggests, is fuzzy and it also spreads. Okay, so they found these 3 kinds of broad phenotypes in a single flask over time. And they show that this WS forms these mats on the surface of the broth. So remember, there's no shaking here, okay? It's just standing there. And the fuzzy spreader kind of forms these clumpy things at the bottom. Okay. Now, typically, this species is an aerobic bacterium. So it's a bit surprising that somebody is even able to grow here at all. So now they're saying, well, what is giving rise to this diversity? Why do we see it? We started off with everything looking like this. How come we end up getting this stuff as well? Right? That's the main question they're asking. What they show in this paper, we won't bother with exactly what is the metric of diversity here. You'll learn more about this in the next few classes. But let's imagine there is some way that we can ask how many kinds of colonies are we seeing in the culture. That diversity increases over time and then kind of goes up and down a little bit, but is increasing in general, right? During the course of this experiment, which is over about 10 days. Now, if they don't allow this to be standing still, if they keep shaking this beaker for that time, it's a flat line, okay? That means that spatial heterogeneity, potentially that's created because of oxygen supply changing in the different parts of that column, the medium column, may be important, because when you shake it, you destroy that and you no longer see diversity evolving. At the same time, they also measure the number of bacteria that exist of each type, each of these 3 types. So the round circles here are the smooth colony, the starting one. As you can see, it grows up very quickly, it's very high, but then it sort of decreases over time. The triangles are the wrinkly spreader, which come up around day 2. Again, they also increase, and then they sort of go up and down a little bit. And the fuzzy spreader is the square, which comes up a little bit later than the wrinkly spreader, again increases and is decreasing later as well. This is kind of interesting. There seem to be some dynamics that are much more interesting than just everybody coming up at the same time. And again, if they shake the culture, they only get the smooth guy. They never see the wrinkly or the fuzzy spreaders, right? So this diversity of these different types is clearly linked to the fact of having a static habitat with the possibility of oxygen being something that's important, that's different. Everybody with me on this? Okay. Okay, so then to sort of further understand and show this effect of spatial heterogeneity, they ask whether the diversity depends on whether we have a static broth, we shake it for some time and then make it static, so your diversity goes back up. But if you keep shaking it, your diversity drops across 21 days, right? So every 7 days now, they're transferring a part of the culture to a fresh beaker with nice nutrients and everything. So this very conclusively shows that that heterogeneity is important for the diversity. And then they do these very nice series of competition assays. What they show here is if we take a full wild WS culture and we put in a little bit of the SM, which is the ancestor, is it able to invade the WS, right? Which means in the presence of a very strong competitor, which is outnumbering me, am I able to replicate faster than this competitor, right? That's the invasion assay in very simple terms. The details are in the paper and you should read it if you have not read it yet. So the arrows are then pointing to the invader and the one that gets invaded. And this number reflects how much better is this guy able to invade relative to who it's invading. So 1 would mean that this and this are equal. So if you start with 99% of this and 1% of this, you would end up with 50% of each, okay, and that would be 1. So 1.66 means that this is about 66% better than the WS, okay, at replicating. So it's able to invade effectively. Similarly, WS is able to invade FS, FS is able to invade SM. This is remind you guys of a game that you perhaps have played. Yeah, it's rock, paper, scissors, right? So effectively, they're saying that all of them are able to invade at least one other phenotype, right? And the only exception is that FS is not able to invade WS. So when there is the wrinkly spreader dominant in the media, FS does not have a chance of invading, okay? But SM can invade it, right? So what they're showing basically is that this invasion, the serial invasion dynamics, and the fact that it is frequency dependent, which means that the frequency of the invader matters. Typically, if the invader is rare, it is able to outcompete the invadee, yeah? That is what they're showing is happening here. So now, so this is what all that they show in this current paper, okay? Does anybody have anything to say about this paper then? So why is this exciting, right? This is the first time you're seeing in such a short term this radiation happening. It's clearly driven by adaptive dynamics because it is adaptive to be a rare new genotype or phenotype. We don't know yet the genotypes in this paper. And each of them are serially selected for in this medium in a very simple microcosm. So those of you who read the paper, was there anything particularly interesting that you found? Just a name for, instead of sort of a real life experiment, right, with a large, it's just a micro, I don't know. Yeah, no, it's an experiment, right? It's just that it's just on a small scale compared to, let's say these real dynamics may happen in a lake outside. Very difficult to sample anything there. So you do it in a small system. Yeah. But please do stop me when I use these kinds of terms which I take for granted. And, yeah.
Can it be a plastic response? Is it heterogeneous response? Is like, you know, you have different environments in local environments, they are showing different response. So is this related to plasticity?
Yeah. So his question is this related to plasticity? How do we know that it is not plasticity? So plasticity is this idea, we talk about it in my next lecture a bit more, where you have the same genotype, but it gives rise to different phenotypes depending on the environmental condition. Is everybody happy with the concept of genotype and phenotype? I think Mridula went over that today, right? Yeah. Anybody who's not still clear? Okay, good. So effectively, plasticity would mean that we don't have, these are not hereditary, okay? These are not heritable phenotypes, but given some specific conditions, SM could become WS without any genetic changes. And thanks for bringing that up, because that's actually something I want to discuss specifically. Why is heritability important? In general, why is heritability important for evolutionary change? So it's carried forward, right? Because the problem with plasticity, in some sense, is that you can't inherit it, right? So it's like Lamarckian evolution in some sense, if you remember. The concept would be that your giraffe's neck gets longer, but then it's no use to its offspring because the offspring have to start from scratch again. Whereas now, in a situation where there are only tall trees in a habitat, and that's reliably going to carry forward, giraffes which have longer necks and are able to pass that trait to their offspring will have an evolutionary advantage. So evolutionary change, which is defined as change in allele frequencies in a population, can only occur if there are alleles to be changed, right? So plastic responses are a very interesting phenomenon of their own accord. Talk about this a little bit more. They can also influence evolution in specific situations, but plastic responses in general cannot directly be inherited, right? So it's important actually to know whether these traits are heritable or not. Yeah? And they show this in this paper to some extent as well. Right, so for any trait under selection, heritability is important, and they do show in this paper that the colony morphology is heritable. Okay. Yeah. I don't recall the details of that experiment. How about I read that paper once again and we can discuss it tomorrow? Yeah. Do you remember? Yeah. Anybody else remembers this work? I'll read it and then we'll get back tomorrow. Yeah. Zula, can you make a note of this, please? I'll add it to my next. Okay. Any other questions, concerns, points you want to bring up? Yeah.
Like in this paper, we have talked about something like frequency dependent selection, right? Like the rare one will have more advantage. But if we see here this comparative thickness from wrinkle to smooth, it's like 2.20. So, but like so in this manner, the wrinkle will keep on increasing. So now this fuzzy one is rare. So now it should have advantage, right? So first of all, I do not know whether they continue the experiment to see whether it's a cyclic pattern. Then only we will say it's frequency dependent.
Yeah.
Because here it can be it's just competitive exclusion. Like the wrinkle one is having more competitive advantage, so it will completely outcompete. Because we have this evidence also when we put fuzzy and wrinkle, its fitness is completely zero.
Yeah.
So in this case, it can be just exclusion by fitness.
Yeah, so it's sort of serial comparative exclusion. So the question is, how do we know it's frequency dependent selection and not just simple comparative exclusion? So it is comparative exclusion in some sense, but it happens serially, right? And it's frequency dependent. So if you start off with
So if we talk about like SM and FS, when we put FS in SM, we know it's having high fitness. And when we put SM in FS, again it's having high. So we know that here it's frequency dependent.
Yeah.
But when we talk about WS and FS,
yeah.
so in that case, when we put FS in WS, its fitness is zero.
Yeah.
So it means it cannot outcompete that. It's not frequency dependent in this case.
In this one case, yeah. So what they're saying there is the reason FS presumably, they don't know the details for this paper. They worked this out a bit later. But they're saying that FS, because it's able to invade SM, and SM is able to invade WS, effectively, once SM gets to invade WS, then FS can invade SM. Right? So that is what they're trying. So it's true that it's not like a complete nice tidy circle. There is a kink. And maybe part of that is explained by some of the subsequent work that I'll discuss in a bit. Yeah. Yeah. Yeah, so no, here in this case, it's clear it's not plasticity because, they, like you said, when you put them back, you know, they retain their phenotype, right? Yeah.
Adaptive plasticity.
No, but when you change the conditions, then the expectation, if it's just plasticity, will go back to whatever the condition is best, right? So for example, if you take a WS, right, it's better in a situation where it's static. But now if you start shaking it, the WS is still WS. It goes extinct until the point where it gets some new mutation and becomes SM. Does that make sense? So they have shown here very clearly in the paper that it is not plasticity, it is hereditary, and that's important. Yeah. Yeah. Not in this paper, no. No. Eventually, yeah, they've done all kinds. I'll show you a movie actually later as well. So I imagine they have, but yeah. Yeah.
Can we argue for the fact that because this FS is only that particular strain which is developing at the bottom of the feature. So in a pretty much anaerobic condition in comparison to the top. So it's not being able to invade WS could be the reason that it's kind of habitat has to.
Yeah, possibly, yeah. So they speculate about this a little bit, but not too much in the paper. I'll come to that a little bit later. So they, yeah, that's probably what's going on. But yeah, we'll see an example.
Because in SM it is all over. So it is
Correct.
easier for them to invade at least the bottom.
Yeah, yeah.
Because that's very
Yep. Okay. So one more question I wanted to bring up is, how would you test what benefits the FS might have? So similar to what you're saying, maybe FS is unable to invade WS because it's an aerobic bug to begin with. It's unlikely that a small set of mutations will make it anaerobic, right, and make it fantastic at growing at the bottom of the culture. How would you test, let's say this aerobic anaerobic is the problem? Fuzzies at the bottom, yeah. Not quite actually, but how would you test this, right? The reason, let's say it's not able to invade, and the reason it's at the bottom, there could be 2 or 3 multiple competing hypotheses, right? One is that it's just, somehow it's a great anaerobe now, because of the mutations it got, and so it's able to happily live at the bottom. How would you test this? Shaking what? And then? Mm-hmm. It can't grow at the bottom once you shake the beaker. Or then leave it again. But then again, oxygen is depleted at the bottom. Give anaerobic conditions, right? Yeah, so somehow make sure that there is no oxygen available at the top, and then ask if the FS is doing better than everybody else. See, the invasion assays are not sort of directly saying spatially where it goes and invades, right? WS is still hanging out only at the top, at the periphery of the culture, of the air broth interface. So WS is spatially never going to go to FS and invade it. Okay, it's just saying that if I take the medium, I allow them to grow as they will, where they will, and then I shake it all up and take a sample and plate out colonies and count how many of each do I find. That's their invasion assay, right? So spatially, FS and WS will always be separate. No, but you haven't provided conditions for it to be anaerobic or not, right? So it's actually a difficult experiment to do. It's not as easy as it seems at first. Well, think about it a bit and we can talk more tomorrow. Yeah, one could still quibble, but yes, this is roughly the kind of experiment one wants to do. Yeah.
Unable to invade in that condition, then we know for sure that after it has some
Good. Okay, all right. So I want to move on to another set of questions now. They also have this statement. So I didn't show you sort of a lot of the variation here, But they've repeated this experiment not once, but many, many times, right? So they've taken sort of 10 or 20, whatever number of beakers, and they keep finding this thing again and again and again. So I have the statement saying that the repeatability of this evolutionary radiation is a hallmark of the fact that it's adaptive, right? That it's selection which is driving this. I wanted to ask you, why do you think repeatability, or how can repeatability be a signature of selection, of strong selection, and under what conditions may that not be the case? Can you think of some conditions when that will not be the case? So the statement they make is repeatability is a signature of strong selection. Right? The fact that repeatably you keep seeing the same phenomenon. So in general, we think this is true. A, why is this true? And B, when will it not be true? Conditions are the same. So then? You would expect repeatability? No, but you do see repeatability. So the question is, if you see something, so let's say tomorrow we do slightly different experiments, right? In a different bacterium, different set of environments, and you see some radiation happening. Question is, whenever a phenomenon is repeatable, when or why does it suggest strong selection versus, well, actually let's talk, what is the alternative, right? What do we mean if it's not selection? Then what's happening? Sorry? Hang on, let's finish one thought. So somebody said drift. Yeah, is everybody familiar with the concept of genetic drift? Anybody who's not? You're not. So the idea here is that something would happen by stochastic events, so chance events, where let's say one particular mutation spreads in a population, not because it is favored, but because the population is very small, and by chance this mutation manages to replicate faster than others which are not, other cells which don't have that mutation. Okay, I think Kavita is going to go more through these concepts later, but that's the general intuitive idea of this. So if drift is the alternative, right? So let's say every time they repeat this experiment, just by chance, they get the same stuff happening, right? So under what conditions would you get the same things happening, same sets of phenotypes evolving, but selection is not driving it? Are there any such conditions? Because if not, then it's a great idea, right? That if you keep seeing repeatable things, then that's a very strong signature of selection. So you're sort of in the right track, but I want you to be a bit more precise.
But the number of outcomes are limited.
The number of outcomes are limited, yeah.
And then again, just by chance, it is, since now it has only a limited amount of outcomes that it can produce.
Good,
So
excellent, yeah.
And then we will think A.
So is everybody with him? Yeah, so if there are only 3 possible phenotypes that could be explored, then they might be explored just by chance, right? Whereas if there are 20 different phenotypes that could be explored and you keep seeing only 3 of them, then it's relatively unlikely that it is chance events. Does that make sense? Because we're saying that effectively the mutations that happen are random, right? So the population as a whole is sampling a whole bunch of mutations, and only some of them are beneficial and will increase in the population under selection. Okay. What else is true? So you said that the conditions are similar, right? Every time they do the beaker experiment, it's the same, right? So what are we assuming here when we say it's the same? That for the bacteria, it is actually the same. There are no micro niche differences between the beakers. So let's say tomorrow I get a very bad set of beakers from a terrible glass blower, or wherever the beakers are made, right? And there are all kinds of sort of pockets in the beaker wall. That's a great niche now, and I might start seeing different phenotypes. Make sense? So when we do these experiments, we assume that our conditions are actually quite nice and homogeneous. Yeah. Okay, so those are sort of 2 possibilities. Yeah. Correct, and the order also is repeatable in this case. Yeah. So in this case, order is also important. They don't talk too much about the order actually, but true. And they make a similar conclusion or similar sort of statement about rapidity, okay, the time scale of this, which is again very similar across the replicates. So again, If it's very rapid that you see this radiation happening, they suggest that it is also a strong signature of selection. Why is that the case? It's related to the previous arguments we made. What do you mean? So a bunch of assumptions that are going in there. And I want you and you to not talk for some time now. Somebody else. What is the assumption that? So did you understand what he was saying? Right? He's saying that effectively if the time is very short, it means the strength of selection is very high. That's why it's able to go to fixation in the population very quickly. What is the assumption here? Okay. Huh? So he's suggesting that the reason that the time in which we see this heterogeneity arising is important. If it's very quick, it suggests it's selection rather than drift. And I'm asking why. And he's suggesting it's because the quick fixation suggests it's very high levels of selection, very strong selection. What are the assumptions that go into this explanation? You haven't learned this yet, so I just want you to think. I mean, in this course, you haven't learned it yet.
Can it be small population and higher rate of reproduction?
Higher rate of reproduction effectively is a metric of selection, right? Or selection coefficient of the new mutations. You're right, the population size has something to do with it, right? So we're assuming that this is only going to be true only when the population size is large enough, right? Then alleles will rapidly go to fixation proportional to the strength of selection, okay? Proportional to the selection coefficient that these alleles carry. So you'll learn more about this in the next few classes, but try and keep this in mind, that population size matters and the strength of selection matters. Yeah. They actually don't start with one cell. They start with many, many cells.
But the one that survives after 7 days,
we don't know in these particular cases because they didn't do any sequencing. This is, I don't know why they didn't, but whatever, they didn't.
same kind of adaptation right from the part.
Yeah, absolutely. So I said they didn't do sequencing here, but they've done a lot more experiments later on and they still continue to. So I'll talk about those a little bit. So we'll come to the genetic basis of things a bit later. Yeah. Okay, all right. So there's a relationship between population size and repeatability and the speed of fixation. So let's say if genome size of these bacteria was much smaller than what it is now, would you predict that you'd see similar repeatability, or are you less likely to see repeatability? If the genome was smaller, just a general guess, right? Less likely, why? Probability of mutation will reduce, why? Sure, but how does mutation happen? Like, how do we measure mutation probability? It's per base pair, per replication or something, right? So the probability of a mutation happening is still going to be the same. More likely, okay, opposite, why? If your genome size is smaller, do you expect the repeatability to be higher or lower? So this goes back to something we discussed earlier, right? That if the number of possibilities you're exploring is lower, there is less likely, more likely that you'll find the same things again and again. Okay, what about if our genetic code of this bacterium was made up of 2 bases instead of 4, then? I'm trying to, by the way, link whatever you've learned from Vidula today morning. That's real stuff, that's useful information to have and to be able to think about it, right? So what if our life was 2 base instead of 4 base? Yeah. Yeah. So you'd find more repeatability, right? And more rapid fixation perhaps. Okay, all right, good. So now in recent work, they've tried to address, yeah. Some mutations will occur just by chance, which have nothing to do with selection, right? And we'll talk about some of such cases later on, that they basically tag along with the good guys and pretend like they're fancy, but they're not. Yeah. Okay. So now I want to talk a little bit about more recent work that these groups have done. Yeah.
When you're shaking it, you're effectively changing the environment, and that environment will be better for a particular.
Correct.
It's a good measure of.
No, you're not doing invasion assays in shaking. No, you're doing invasion assays in static broth. If they shake, they only get the smooth guy, and nobody else is coming up. Because the WS and the FS are both dependent on the static condition. Yeah? Okay, why don't you go and read the paper and then we'll talk again. Any other questions? All right. So I want to talk about recent work where they've tried to figure out what is the genetic basis, right? What is with this FS and WS and what mutations are there, how many mutations and so on. So in general, I'm just summarizing a bunch of different work they've done. Here's a set of genes that they figured out in the genome, okay, of this bacterium. This is standard notation sort of show genes. This is the start of the gene, this is the end of the gene and so on, okay? And these genes are arranged in bacteria very often in clusters called operons. And they're related, usually performing related functions. So what they find interestingly is that the WS and FS mutations that are responsible for these phenotypes are all happening in the same gene, which is part of this operon, okay? That itself is interesting, right? Because they're completely different phenotypes, but they arise from mutations in the same gene. The interesting thing is the mutations are in different parts of the gene, okay? Now WS leads to, in general, the overproduction of cellulose. Cellulose is something which will help this mat to form. It's a tight binding, right, at this interface of the broth and the air. And FS mutations end up altering the surface lipopolysaccharide, which is something that bacterial cells secrete, which is sticky substance. Again, helps them to form some sort of raft. So different mutations in the same gene give rise to distinct phenotypes. That's one important takeaway from this work. WS is also obtained by mutations in other genes. This is not the only gene responsible. Okay, so they sequenced a whole lot of different WS phenotypes from different experiments, and they found that in some cases, this gene is responsible. In other cases, other genes are responsible, all of which eventually feed into the cellulose production pathway. Okay, so a second take home is that similar phenotypes can be obtained from mutations in different genes. Yeah. And finally, what's really interesting is that WS forms thick stable mats, but FS forms flimsy rafts that drop like snowflakes. And this they figured out when they did some nice time-lapse photography or videos of these films and so on. Okay, so I'm going to show you that on the next slide. Okay, so I don't know if you can see very well, but so this guy has gone cloudy, and the FS, yeah, see there are these things that are formed and then they drop. Things that are formed and then they drop. And then WS has this very nice thick layer that's formed always on top. Can you turn off the lights? Let's go once again. Able to see? So what they're basically then showing in this work is that the FS is interesting. It's actually linked to WS, the evolution of FS and WS, because they're sort of in the same set of genes. And FS, this is why FS can't invade WS, is because it's too flimsy and it drops, right? So then it has no access to air and it sort of manages to do something. So that's why it's also better able to invade SM, is because it blocks SM from having access to air as much, to oxygen as much when it forms these rafts. Right? So that's the idea, and that's why WS is such a champion. Just hangs on. And they did these really fun assays where they put glass beads of known weight on top and asked whether WS or FS, how much glass bead basically they can hold, and WS is a champion weightlifter. It hangs on to these glass beads for much longer and much more glass beads compared to the FS, which just collapses under its own weight very, very quickly. Yeah? So this is kind of where I want to stop with this work. And hopefully this connects and helps you connect well with the next set of talks and lectures that you'll hear from tomorrow onwards, which will dive much deeper into mutation and selection and the interaction between population size and all of these different processes. Yeah? I suggest that you take your reading seriously for the journal club discussion, because it's much more valuable and more fun if people have read it and thought about it. And of course, today's was a little bit difficult because background and don't quite know what to expect from these. But do read and think about the next set of papers that are assigned for each journal club, and come prepared with some questions or something that you did not and here, and we can then have better discussion. Yeah. Any questions for today? Yeah. I don't understand. Yeah, true. No, so what they've done is they've done serial transfers, right? Where they've gone from one beaker to the other beaker, and they sort of have to do that because the nutrient runs out at some point, and then nobody can grow. So to keep these dynamics and to observe them over time, they have to keep transferring to new flasks. So, yeah. I'm not sure whether there's a question there. I'm confused. Huh? Yeah. Uh-huh. Ah. Mm-hmm. Yeah, no, good points to note. Yes, I agree. And this is interesting to follow up for sure. I think we might come to this a bit later, and some examples we'll look at in the next few lectures. But so, okay, so then let me just reiterate the 2 points that she's trying to make is, you might see diversity earlier, but we expect that if selection is directional and is continuous, we'll weed out some of these variants over time. In this case, it's happening because of frequency dependence. New variants come up and displace the older variants, right? And with sustained selection, you want to be sure that this variability is meaningful and is retained. In general, unless there is frequency-dependent selection happening or diversifying selection happening, you will not see much variation exist in the population over longer-term periods, right? Because you expect selection to throw out the ones that are not as fit. And then the second point is that selection is important in the heterogeneous environment that maintains diversity. So actually that makes it a third point, third way in which you can retain variability over long-term periods is if you have such heterogeneity, right? Either in terms of spatial or temporal heterogeneity. And in some sense, frequency-dependent selection is kind of temporal heterogeneity, right? At different points of time, you expect that the fitness of a particular morph is different. Okay, good. Any other points or questions? Yeah. No, allopatry may also be, yeah. So adaptive radiations do not really say anything about whether it occurred in sympatry or allopatry. No, not really. Ah. Not just geographical, yeah, geographical, yes, you're right, yeah. The radiation, yeah. I mean, they may not, right? They may be able to mate or they may be able to mate with across different populations for a long time afterwards. And there are known cases of that as well, where there is complete absence of gene flow. And yet, if you bring those populations together after whatever thousands of years, they're still perfectly fine and able to produce fertile offspring and so on. So it's not guaranteed that allopatry will always give rise to speciation. In general, we think that allopatry is more likely to allow speciation to occur compared to sympatry. So by that logic, in general, radiation should also be more likely under allopatry compared to sympatry. I mean, in general, if you just count up, there are relatively few known cases of sympatric speciation in general compared to allopatric speciation, for which we have hundreds of examples. Yep. All right, great. So I hope you had a productive day and lots to think about. See you tomorrow.